Hey everybody, welcome back to Bio 233 lecture series. We just finished urinary and we're about to start fluids, electrolytes, and acid-base balance. This will be a couple lectures. We'll break this up into probably 2 lectures to talk about all the different topics. So why don't we go ahead and get started looking at the lecture notes. So the first thing is we want to talk about, if we're going to talk about fluids and electrolytes, we need to talk about the fluids. So we're going to talk about water. And water is our solvent, right? The thing that is almost all of our dissolved substances in our body are in water. And water makes up a huge portion of us. Your water content varies depending on your age. But can generally be somewhere to a little below 50% to up to around 70-75% in infants of total body weight. One reason for that is that the changes is that fat has less body water. It's the least hydrated of tissues, and muscle has a lot of body water. So unfortunately, as we get older, we start to lose some muscle mass, and so our body water starts to drop a little bit. So there's that. Now, where is this water in our body? Where do we find it? And so this picture here, there's a sideways version of this picture, which is pretty much the exact same thing on 1013. I'll show it to you here in the book. And what this shows is the total volume of water. And they're using kind of an adult male that's 150 pounds approximately. So there we go. And then it breaks down, and it breaks it down into 2, extracellular fluid and intracellular fluid. Intracellular fluid is basically cytoplasm, right? And this is all inside the cells. And this is where the majority of the water is. About 40% of your body weight, or about 2/3 of the total body water, is here. And then we have extracellular fluid that's in your body but outside of the cells. And we find that in 2 places. So we have interstitial fluid, that thin layer of fluid that kind of surrounds all of our tissues and our cells. And when you add up that thin layer, it actually gets to be a significant amount, about 12 liters in your body. And then for plasma, it's about half of the volume of blood, and blood is normally around 5 to 6 liters, and so we're looking at about 3 liters of plasma. Those 2 make up the extracellular fluid. Okay, so that's what this says down here, and you can see that nice little picture. So we say intracellular. We sometimes also use the term intercellular. Right, but intra or intercellular are basically the same. And so this is within the cells, and about 25 liters. And as I said before, this is a good way to think about this is cytoplasm. And then we have extracellular. Actually, so hold on, let's do this. So we have intracellular here, and we have extracellular there. And as you can see, we can use the same colors here. We have interstitial fluid, which ends up being about the majority of the extracellular fluid, and this is the fluid surrounding the cells and between tissues. This also includes cerebrospinal fluid, lymph, fluids of the eyes, ear, GI tract. Anywhere there's fluid that's not inside a cell, except for blood, is this. And our blood fluid, blood plasma, is intravascular, 3 to 5 liters, depending on the size of you, within the blood vessels themselves. Okay? So there you go. So now that we know where the fluid is, let's start talking about what's inside those fluids. And so we want to look at the solutes, right? Solutes are dissolved substances within a solvent, and that solvent is water for us mostly. And so we want to look at electrolytes and non-electrolytes. What are those? So electrolytes have ionic bonds that dissociate in body fluids, and the ions are either negative anions or positive cations. So if we think of a negative anion, we might think of a chloride ion there, right? We want to think of a positive cation, we might think of a sodium ion. Okay, and all acids, bases, and salts are electrolytes. So all of them dissociate in the water, and when they dissociate, they contribute 2 or more particles. Okay, and so they're showing a couple examples here. Note this 2, CaCl2. We should really write that correctly, right? I don't know why it didn't lowercase there. CaCl2, the 2 in a subscript. And what these are showing, these electrolytes, is when they dissolve, we get a cation and an anion, or in this case, 1 cation and 2 anions. Okay, and they now can, since they're dissociated, they are free to move apart from each other and move separately, and that's what it means at the bottom. If it's important if they move from one compartment to another, because the sodium may move by itself, the sodium might move and the chloride might follow. The chloride might be, because it's negatively charged, attracted to something else and held on to it. So we can use the parts of the electrolytes as needed. Now, non-electrolytes have covalent bonds, right? So these are bonds that don't dissociate very easily in solution, and they don't have charge. No charge, and so remember that is a way to say that they are, we can just put it over here, that they are non-polar, right? They don't have charge. And many organic molecules fit in this. So most of the organic molecules that we're going to break down and use as food, and a lot of the organic molecules that are waste products, are going to be in this category. Okay. So comparing solutes, plasma and interstitial fluid are pretty similar, meaning that what is in one is usually in the other, and this is because things can dissolve, or can not dissolve, but can diffuse out of the plasma into the interstitial fluid and vice versa. However, there's big differences between what's in the intracellular fluid and the extracellular fluid, and that's what this graph here shows. And this is not something that you need to memorize, but what I'll highlight here real quickly, and then we can show in the book, is the extracellular fluid has some specific electrolytes that it has a lot of, and the big player is sodium, right? Now we also see some of these other ones, like chloride, makes sense since it's coming from sodium chloride, calcium, and then bicarbonate ion. It also contains a lot of protein anions in plasma, but outside of plasma, not much more. So plasma contains more protein ions, all right? But overall, there are more proteins in the intracellular than the extracellular fluid. So let's make a note of that, and let's look at that in just a minute. Now, proteins usually have charge because the amino acids have different R groups, and those R groups can have charge. So that's why we're including proteins into these solutes, because they have charge and they dissolve in water. Now, if we look at intracellular fluid, and by the way, there's a typo here, so we want to fix that typo. This is magnesium. Ma is not a atom. We want to look at, we can do it in a different color, just for, we have a different color, and the big player is potassium, and sodium and potassium are usually in different locations in the body. We've already seen this with nervous system and muscle cells, where we have sodium on one side of the membrane and potassium on the other side of the membrane when we are in a resting potential, and then they switch places. And we see this in a lot of places in our body. The positive charged sodium tends to be in extracellular fluid, and the positive charged Potassium tends to be an intracellular fluid more. So now let's just, you don't have to memorize this graph, but I do just want to show it to you very quickly so you can get an idea of what it's trying to say here. So if we look at the red and the blue lines, that's extracellular fluid, right? Fluid out, not inside our cells. The red is blood plasma, and the blue is just other interstitial fluid, okay? And so if you look, they're very similar. This is the concentrations, milliequivalent per liter, all right? And so they're looking at here sodium, so a lot of sodium in extracellular fluid, not much in intracellular fluid. And then look at the switch, very little potassium in intracellular fluid, very little potassium in extracellular fluid, but a lot of potassium in intracellular fluid. And then, as I said, there's some others that are players here in extracellular. There's a little bit of calcium, hardly any inside. There's a decent amount of bicarbonate, and there's a lot of chloride, right? And some protein anions in blood, particularly. When we look at intracellular fluid, we see lots of potassium, as we said, some magnesium, some phosphate ions as well, which we didn't really mention there. I mean, they're on the page, but I didn't mention them. And then a lot of protein anions. And this makes sense because there's a lot of enzymes inside our cells. Remember, this is intracellular fluid. This is in the cell cytoplasm. So there's a lot of proteins that are enzymes and are doing things inside the cell that have purpose there. So again, you don't have to necessarily memorize this entire chart, but it is useful to know which are the main players inside cells, which are the main players outside of cells, and get a little understanding of that. Okay. All right, so now let's talk about movement of fluid between compartments. And I'm going to just say some basic things here because some of this has been, you've done already, and this is just kind of a way to think about what you've already learned. So there's an ongoing exchange between fluids between compartments. And if there's solute concentration somewhere that's changing, water tends to change too because water wants to dilute solutes. So if water can move, it will move from one compartment to another. The extracellular fluid solute concentration is the major regulating factor for both. And that makes sense because blood plasma is getting nutrients and liquid from our digestive system, and then it's transferring that from the blood into interstitial fluid, and then it's transferring what needs to go from there into the cells. So plasma and interstitial fluid solutes can move in both directions, and this is using capillary dynamics. And so the only thing I'm going to say here is review, right? You need to know this for the final. So we want to think about how do we move those things around there. In kidney filtration, there is different pressures there that move fluids around and the solutes as well. And so you want to review that also. We're not going to go over it now. You're going to review it for your final exam. All right, now between the interstitial fluid and the intracellular fluid, it's a lot more complex. And to the point where we're not going to really go into a lot of detail because you kind of have to look at each tissue and each cell and see what it's wanting and needing. But generally speaking, we can say a couple things, okay? So again, interstitial fluid, remember, is extracellular fluid. Intracellular fluid is ICF, right? So from the interstitial fluid, which is outside of the cell, and the intracellular fluid inside the cell, lots of changes. Usually we will say that it's unidirectional for ions. So if you have a lot of sodium outside, if it is going to move, it's going to move from outside to inside because of the concentration gradient, right, for example. So when it does move, it'll move in that direction. If it's moving passively. Movement of nutrients, glucose, and respiratory gases in and out are going to depend on demand, right? This is going to be down, move via concentration gradients. And one way to think about that is demand. So if you have low oxygen somewhere and you need to have more oxygen and you have higher oxygen in the blood, oxygen will diffuse out, right? So we've talked about that a little in 232. Water can move in both directions, and it's usually dependent on sodium outside the cell and potassium inside the cell. But then also, don't forget, it relies on our two hormones for water movement as well. Whether we're going to bring water into the blood, we're going to put it out into the urine. And we're going to talk about this a little bit later, so I'm not even going to comment on it yet. It'll be on the next page, but we'll get there. So let's look at water balance for a minute. This picture is on page, one second here, I have to look in my notes, because it says a page number and I think it's wrong. It's page 1016, and actually I don't know if I need to go to it because it looks almost exactly like this. Colors are the same, everything looks pretty similar. So this is showing us the intake and the output of water per day, and generally speaking those should balance, right? The amount you take in and the amount you remove from the body should be about equal. If not, then you're going to have increased water weight, or you're going to start to get dehydrated. So a couple things really quickly before let's look at the intake here. We start with the intake. So most is coming and this is for humans, of course, most is coming from beverages, water, things that have water as its base. About 60% of the water we take in comes from beverages. About 30% is water inside the foods that we eat. And then we make water, about 10% of our own water. Remember, when we break down glucose, some of the waste products are carbon dioxide and water. So we're actually producing water by going through metabolism. Okay, so that adds up to about 2,500 mils. Now we'll go over to output it in just a minute. It's on the next page. Let's just highlight a couple things here. So we know that intake is due to thirst mechanisms, right? Stimuli to tell us that we're thirsty or not. And a decrease in plasma fluid volume by about 10%, or osmolality of 1 to 2%, that means the amount of dissolved substances in water goes up about 1 to 2%, will cause the process of the hypothalamus to start to tell us that we're thirsty and to have a seek out liquid. Most quenching and thirst inhibition, and so inhibiting this process, is once we get distension of the intestine, water absorption, and then we lower osmolarity. So this is a negative feedback loop, right? So we trigger the negative feedback loop here, and we end the negative feedback loop with those processes. All right, so now output are listed here, but let's go ahead and look at them on this. So the average output a day, so about 1,500 mils is urine a day. About the same amount we take in beverages. So that's the majority. Then we have insensible losses via skin and lungs. And this means that every time you breathe out, there's some water vapor in your breath. Right now, even if your skin is dry to the touch, very small amounts of water are being lost through your sweat pores, sweat glands, and onto the skin. Those are insensible losses. Now, regular sweat which you actually feel and you can tell that you're sweating, that's different than what we're referring to here, and that's about 8%. And then fecal matter has a little bit of water in it still, around 4% of the water. So the majority of water we get out through our urine, which is probably not a surprise to you. And that's what this talks about there. So all of those things are highlighted. The insensible, ones we don't notice, ones we do notice. Okay. Urine production is normally about 1,500 mils. It can go down, especially if we need to conserve fluid. So if we're dehydrated, we can produce as little water in our urine, or put as little water in our urine as possible. We need some because we're getting rid of toxic waste products and we need to dilute those. And we've already talked about some of the fluid loss things that you could think of when we were talking about like hypovolemic shock. Loss of fluids can be due to vomiting, diarrhea, excessive skin burns, increased blood pressure, changes in diet. All sorts of things can change fluid amounts. Okay, now what I want to do is I want to spend a little bit of time talking about edema, dehydration, and a few other things. And I'm just looking here at the notes, and I think what we'll do is we'll go all the way to the next page, and then we'll take a little pause there and continue on. So disorders. So first, dehydration. This is a negative water balance where the water loss is greater than the intake. So if all that means is this side of this graph, we're losing more water than we're taking in. So if we are getting rid of 2,500 but only took in 500 in beverages, we would be losing more water than we're taking in a day, and we're going to slowly get dehydrated. So that's called a negative water balance. So what happens in this case is we lose water from the cell to the interstitium to the plasma because we don't want our blood volume to go so low that we pass out. So if we start losing water, we need to move that water from our cells into the interstitium, and then water will move from the interstitium to the plasma, and that will keep our blood volume high. And that's what we're seeing down here in this picture of the mechanism of dehydration. Cells lose water to the extracellular fluid by osmosis, and the cells start to shrink, right, as water gets poured in. They're using a little faucet here to be kind of funny. And then water in the interstitial fluid will move into the plasma. So let's type in those because it doesn't actually say. So this is plasma. This is, interstitial fluid. And these are body cells, right? And then guess what? Same thing here. So we have plasma, interstitial fluid, and body cells. Okay. Okay, so causes water loss. Like we just said, anything that causes water loss can cause dehydration. So let's just underline all those things that we kind of underlined last time when we talked about hypovolemic losses back in 232. So what are the signs and symptoms? Well, obviously low urine production, oliguria, or really the amount of the urine is low, and it's also very dark in color. Dry mouth, thirst, dry or flushed skin. Now, one of the treatments is salt pills. And so the question is, why would you give salt when you don't have enough water? Now, of course, you're going to try to rehydrate the person by giving water, right? If we know someone's dehydrated, we want to give water. But often we give saline or salt pills along with it because the water, remember how we were talking about with urinary system, water likes to follow salt. So if we take in some salt into our blood, then we will, and then we hydrate, water will want to dilute that salt and want to stay there and not go out the urinary tract and out into our urine. So one of the main things is because water follows salt and wants to dilute it. And when I say words like follows and wants, it doesn't mean water actually has any opinion about this. What I mean is that there are chemical and physical properties of water and salt that make this happen. So that's one reason why we would want to do that. And that would help get our blood volume up and keep water in the blood. And then as we hydrate, more water can leave the blood and go back into the cells and make those cells work better. All right, so now I want to talk about another kind of the opposite of dehydration, and this is called hypotonic hydration, right? So hypotonic is where you have a lot of water and little sodium. And this actually is called a bunch of different things. Dilutional hyponatremia. I've actually seen it called delusional hyponatremia as well, because it can cause mental confusion, or water intoxication. And that sounds kind of funny, because most people would say, well, water isn't act as a poison, but too much water can cause our body not to work correctly. So you can become intoxicated by water. And what we're seeing when we see this is we have so much water that we have very low sodium levels. Okay? So we're either for some reason having sodium levels be not reabsorbed, all right? Being not, there's a word missing here. Let me try to, I'm going to just type it in. I can't get to the exact spot I want. Either excess sodium not being reabsorbed, or excess sodium being excreted by the kidney, so we're getting rid of sodium. That's kind of one possibility. Or the ratio is low due to excess water. Because if we have a lot of water, that just by default dilutes all the solutes. All right. And so when we have excess water, and this is usually the more common one, is that we have then a lot of fluid goes into the cells. So if you look over here, that's what we're seeing for this one, this hypotonic hydration. We have too much water and less sodium. And so that water wants to then go away and make the sodium levels correct in the plasma. So the water goes and enters the interstitial fluid. And then that water goes into the body cells, and those cells will start to swell, and they can start to not work correctly. One thing that's always problematic is we have too much water, then we swell things and we push things away from each other, and it takes longer for nutrients to go from one side of the cell to the other, to go from the blood into the interstitium, into the cell. And too much water actually slows all those processes down and can cause lots of problems. All right. So what are the ways that we might have excess water? Well, one is renal insufficiency. Our kidneys aren't working correctly and water's being retained. The other one is ingestion of large amounts of water in a short period of time. This is temporary but could be very serious. Sometimes, I'll give you a quick example. Sometimes like marathon runners or long distance runners think that they're sweating a lot, and so as a result they are drinking a lot of water. But sometimes because they're kind of overworking their body, their kidneys actually shut down and they aren't sweating as much as they think, and they keep consuming large amounts of water, and what they're doing is they're diluting their sodium levels. And once you start diluting your sodium levels, think about what will happen. Where do we use sodium? That's so important. Nervous system, right? All of our firing of neurons. Muscle system, because we have to have action potentials go across our muscles. And so as a result, you're going to see a lot of problems in those areas, like muscle weakness, headaches, not hypotension, we have hypertension. Oh, I know why, hypotension. Because some of it might leave the plasma and go in, and so fluid is being retained here, but less so. Tachycardia, we see. Circulatory shock, we can sometimes see if hypotension gets really low. And if it's really severe, we get mental confusion, stupor, and even coma. And you can die from this. It has happened before where people have ingested too much water, become too highly water intoxicated, and have died. So what's the treatment? Well, we actually give more water, but what we give is not just regular water. We give a hypertonic infusion, and that is so we put a lot of solutes in. So if we're really low on sodium, we bring water in that has lots of sodium, and that makes the sodium level balance out. Okay, so those are our two: lots of water, too little water. And then we already mentioned, but we're just going to kind of remind ourselves about it, is edema. And we talked about it being atypical accumulation of excess interstitial fluid, swelling within tissues. This is all review from when we looked at this in capillary dynamics, and this is the same thing as what I was talking about here about having too much water. We get too much fluid in the interstitial spaces, which swells them, and this may impair tissue function due to the distance between the capillaries and cells. Oxygen and nutrients can't reach cells as quickly. They probably will reach them eventually, but as quickly as they should, and the cells can be impaired. Okay, so causes of edema. Any event that enhances movement of fluids out of the vessels and into the tissues or causes backup of the blood into the capillaries will cause edema. And so you might remember, we kind of went over some of these before. Increasing capillary permeability, like allergic responses, will cause more water to come in. Elevated intracapillary pressure, hydrostatic pressure of the capillary. That is our friend HPC. So you can go back and remind yourself about that. That could be due to arterial dilation. That could be due to blocking veins, and so water can't flow through, and so it starts to build up pressure in the capillary bed. It could be due to increased venous pressure, which makes it harder then to push water out of the way, or increased flow to capillaries. If we did, that's similar to, that might be tied in a little bit to the capillary permeability as well, bringing more fluid in and more blood flow into capillaries. Anything that increases the pressure will increase filtration rate. So there's a missing arrow here. Anything that increases pressure will increase filtration rate. So if blood pressure goes up, the filtration rate goes up from plasma to the interstitium. Now we also have, what if our lymphatics are blocked? So what if our lymphatics are blocked and we can't get fluid out of our interstitium and through our lymph system? Well, that increases our interstitial fluid colloid osmotic pressure. That one is our OPI. That goes up. Okay, so this might be due to lots of proteins in the interstitial space. If we have a lot of proteins that have spilled into the interstitium due to the result of a trauma or a blister or something like that, then we're going to have a lot of water go in to try to dilute them. That's another way that will increase OPI. And then the last one is we can have decreased plasma colloid osmotic pressure in the capillary, and that is OPC, osmotic pressure of the capillary. And this is due, if you remember, to loss. Here, let's do a different color because we're doing each one of these in a different color. This is due to loss of excess protein in the urine. Okay, so if proteins that are in our blood, they keep their anions, they are dissolved in water, and they keep fluid in our bloodstream. If we lose those proteins, like they go out in our urine because of not working glomeruli of our kidneys, then we're going to have less proteins in our blood, and therefore water won't stay in our blood as much, and water will go out. And so that will go out to the tissues, and that will cause edema. Okay? And with long-term edema, there could be lowered blood volume because of this water moving from the blood into the fluids, into the tissues, and lead to a decrease in blood pressure. Okay? This little diagram here, or this little thing, is just kind of a little summary of what we talked about and how fluid moves from plasma to interstitium to cells to the lymph. So it's a good little review, and you can go ahead and read it. All right, what I think we're going to do is we're going to stop there, and we're going to talk about electrolyte balance and acid-base balance on our next talk. So this is a good place to pause. So review some of that stuff. We went over some things that we talked about in 232, some of the things that we talked about last week in urinary, and we're kind of piecing them all together, and we're trying to make some sense between things like capillary dynamics, edema, fluid movement, and solute concentration, and we're trying to kind of put it all together. So review those things if you need to. Go back and read your notes and tie all this into a nice, neat package. Okay, great. Thanks.